Hello everyone, I am Dr. Trupti and you are watching my YouTube channel Enjoy Biochemistry. In today's video, let's learn the biochemistry practical, that is estimation of serum uric acid by Uricase-PAP method. What is uric acid? Uric acid is the end product of purine metabolism. Purines are adenine and guanine. So let's see how uric acid is formed in the body in brief. Adenosine monophosphate is converted to ionosine monophosphate by the action of enzyme adenosine deaminase. So ammonia is released in this reaction. Then this ionosine monophosphate is converted to ionosine by nucleotidase. Further, this ionosine is converted to hypoxanthine by release of ribose 1-phosphate by the action of enzyme purine nucleoside phosphorylase. The next step is catalyzed by xanthine oxidase and there is formation of xanthine from hypoxanthine, and the last step is formation of uric acid by the action of enzyme xanthine oxidase. Uric acid can also be formed from guanosine monophosphate. So by the action of enzyme nucleotidase, guanosine is formed. Then from the guanosine, guanine is formed by the release of ribose 1-phosphate by the action of enzyme purine nucleoside phosphorylase, and there is release of ammonia to form xanthine, and further by the action of xanthine oxidase uric acid is formed. So that's how in the body purines are catabolized to form uric acid, and uric acid is normally excreted in the urine. The normal range of uric acid in males is about 3 to 7 mg per deciliter, and in females it is 2 to 6 mg per deciliter. You can see slight variations in different textbook or different kits that we use in the laboratory. What is hyperuricemia? When there is increased uric acid level more than 7 mg per deciliter in males and more than 6 mg per deciliter in females, it is called as hyperuricemia. In some books it is mentioned that more than 6.8 mg per deciliter, then it is called as hyperuricemia. At physiological pH of 7.4 and temperature 37 degrees centigrade, uric acid occurs as urations, that is the ionized form of uric acid, and in the extracellular fluid the monosodium urate crystals are formed because the sodium is major cation present in the extracellular fluid, so urate is present as a monosodium urate and it is minimally soluble. So ECF, that is extracellular fluid, is easily saturated with the monosodium urate just above the normal level. So if there is hyperuricemia, there is tendency to form the crystals, and these crystals get precipitated and get deposited in the synovial fluid in the joints, leading to the gouty arthritis. So the manifestations are due to low solubility of this uric acid, and the more precipitation of crystals is due to the low temperature and low pH. So as the temperature decreases and pH decreases, there is more formation of crystals. As this is a case-based quantitative estimation, we always start with the case. A 58-year-old retired Major General complained severe pain in joints and swelling of base of first metatarsophalangeal joint. He was a non-vegetarian and chronic alcoholic. He denies any trauma to the big toe. On examination, he had mild fever and the right big toe was swollen, warm, red. Mention the probable diagnosis in the patient. So what is the probable diagnosis in the condition? It is the case of gout. Now what is the justification for this diagnosis? The key points given in the clinical case, that is swelling and severe pain at the base of first metatarsophalangeal joint, and in the gout the first metatarsophalangeal joint is affected in 90% of cases of gout. So it is the characteristic feature, and gout is more common in non-vegetarians, and chronic alcoholism is also precipitating factor for gout, and moreover there is no history of trauma to the right big toe. So all these key points which are given in the clinical case, they are suggesting or pointing towards the diagnosis of gout. Now the most common parameter that is deranged or associated with this condition is uric acid. So the question is estimate the biochemical parameter in the given serum sample to support the diagnosis. So obviously we are going to estimate uric acid level in the serum. So the name of experiment here is estimation of serum uric acid by Uricase-PAP method. You should always mention the name of experiment and name of method. The method is Uricase-PAP method. Now the next is we have to write the principle. So what is the principle here? The principle is the enzyme uricase. It converts uric acid present in the serum sample into allantoin and hydrogen peroxide. Now this hydrogen peroxide, which is formed in the first reaction, that is by the action of uricase, it further reacts with phenol and 4-amino antipyrine in presence of peroxidase enzyme to form red colored quinone dye, and the intensity of color formed is directly proportional to the amount of uric acid present in the sample. So this is the principle of estimation of uric acid by Uricase-PAP method. Now let's see the procedure. For this procedure we need reagent. So we have single reagent, that is the enzyme reagent. We have serum specimen and uric acid standard. So this is the single reagent of uric acid, and uric acid standard having the concentration 6 mg per deciliter. Why do we use standard? Because standard is a solution of known concentration. So it is like a reference and we can find out the concentration of unknown by comparing this with the standard. And the procedure is we take 3 test tube, blank, standard, unknown, and in the 3 test tube we add 1 ml of enzyme reagent, uric acid reagent, 1 ml in each test tube, 20 microliter of distilled water in the blank, 20 microliter of standard in the standard test tube, and 20 microliter of serum in the unknown test tube. 20 microliter means 0.02 ml. Then we have to mix it, incubate for 5 minutes at 37 degrees centigrade or 10 minutes at room temperature, and we get such color. So you can see here 3 test tubes, blank, standard and unknown. We know that the standard concentration is 6 mg per deciliter, and the color, that is intensity of color, that is developed in the unknown is means the intensity is more as compared to the standard. So by looking just at the color we can say that the concentration of uric acid in the given sample is definitely more than 6 mg per deciliter, and after the incubation we have to read absorbance at 520 nanometer. Now we get ODs, that is OD of blank 0.01, OD of standard 0.12 and OD of unknown is 0.24. Now we will write the formula, that is concentration of uric acid in the serum in milligram per deciliter is equal to OD of unknown minus OD of blank divided by OD of standard minus OD of blank multiplied by concentration of standard in milligram per ml multiplied by volume of standard in ml, and that is multiplied by 100 divided by volume of sample. Everything here is in the ml. So by putting the values, that is concentration of standard is 0.06, volume that we have taken is 0.02, and it is multiplied by 100 divided by 0.02, and we get the multiplication factor 6, and then we put the values 0.24 minus 0.01 divided by 0.12 minus 0.01 multiplied by 6, so we get the value 12.5 mg per deciliter. So the result is the concentration of serum uric acid in the given sample is 12.5 mg per deciliter. Now we have to write the normal range. The normal range, in males it is 3 to 7 mg per deciliter, in female it is 2 to 6 mg per deciliter. So definitely it is increased. So this is the case of hyperuricemia. Now let's see the various causes of hyperuricemia. Hyperuricemia means increased uric acid concentration. So the causes of hyperuricemia are mainly due to either increased formation of uric acid or decreased excretion of uric acid or combination of both. So the first cause is gout. Gout can be primary or secondary. The primary gout is a result of inborn errors of metabolism and the metabolic disorders that mostly occur due to inherited enzyme defect resulting in increased synthesis of purine nucleotides leading to marked increase in the uric acid and further gout. And the secondary causes, they are the manifestation due to certain clinical condition causing increased synthesis or decreased excretion of uric acid. So we will see this gout in detail in the separate video on gout. Then the other condition associated with hyperuricemia is Lesch-Nyhan syndrome. It is due to the deficiency of enzyme HGPRTase, that is hypoxanthine guanine phosphoribosyl transferase, and this is the enzyme of purine salvage pathway. So if this enzyme is defective, there is decreased salvage pathway and there is more accumulation of phosphoribosyl pyrophosphate. So there will be more purine production and more catabolism, leading to formation of uric acid. The another disease is von Gogh disease. Von Gogh disease is the glycogen storage disease, which is due to the deficiency of enzyme glucose 6-phosphatase. So here glucose 6-phosphatase cannot be converted to glucose. So there is accumulation of glucose 6-phosphate, which is diverted to the HMP pathway. So there is increased formation of ribose sugar, more formation of PRPP, increased purine synthesis, increased catabolism, and lastly there will be increased uric acid. So von Gogh disease is associated with hyperuricemia. The conditions like leukemia, polycythemia, they are also associated with the hyperuricemia. Because of rapidly growing cells, there is increased turnover of nucleic acid. Hyperuricemia is also associated with the lactic acidosis. Because this lactic acidosis, the lactate competes with the uric acid for tubular secretion, and whenever there is increased lactic acid, so that acidosis will lead to decreased pH, and decreased pH favors the precipitation of urate crystals. So that's how the lactic acidosis is associated with hyperuricemia. Diabetic ketoacidosis, there are increased ketone bodies, and ketone bodies also compete with uric acid for tubular secretion, and that's why there is hyperuricemia. In conditions of glomerulonephritis, there is decreased GFR and decreased uric acid secretion. So all these conditions are associated with the hyperuricemia. Now let's see some causes of hypouricemia, that is decreased uric acid. So first cause is xanthinuria, that is excretion of xanthin in the urine. It is due to deficiency of enzyme xanthin oxidase, which converts xanthin to uric acid. So there is decreased uric acid formation in this condition. In case of severe liver disease, there is decreased uric acid formation, so there is hypouricemia. In Fanconi syndrome, there is defective renal tubular reabsorption, so there is decreased renal tubular reabsorption of uric acid, and that's why there is increased excretion of uric acid and it leads to hypouricemia. Wilson disease is due to deposition of copper in the hepatocytes, and that's why there is decreased uric acid formation. So these four important conditions are associated with the hypouricemia. So now let's correlate our finding with the clinical case scenario. So we got the result, that is the concentration of serum uric acid in the given sample is 12.5 mg per deciliter. We know the normal range means it is increased, so there is hyperuricemia. So probable diagnosis was gout, and for that the key points in clinical case mentioned were swelling and severe pain at the base of first metatarsophalangeal joint, which is very common in gout, and non-vegetarian diet and alcohol. These are the precipitating factors. Moreover, there was no history of trauma. Now on lab investigation, we found that serum uric acid level is also increased. So it is correlating with the diagnosis of gout. The most likely questions that can be asked on this clinical case are: mention the various causes of gout. What are the other clinical features seen in this condition? Explain the biochemical basis of gouty arthritis in alcoholics as well as the people consuming non-veg diet. How can the patient be managed by the clinician, and what dietary changes would you suggest to the patient? So we are going to discuss the answers of all these questions in the next video, that is the video on gout, a case study. So I hope this video on practical biochemistry, that is estimation of serum uric acid, will be useful to you. Thank you for watching and happy learning.